Okay, so this is one of a series of webinars that I've been presenting regarding Alvetex technology, and it's a technology that enables you to grow your cells in 3 dimensions in vitro. And today's focus is on a neuroscience application, so 3-dimensional culture of cells and their differentiation into neural tissues. So the way I'm going to do this is, first of all, give you an overview of 3D culture and introduce Alvetex technology. So that's quite generic to all the applications, and then talk specifically about its use for neural, and then finish off with methods that are compatible with using this technology. Okay, so what we have here is the normal situation in vivo. So this is tissue shown histologically, and we can also then show that schematically. So you see individual cells with extracellular matrix. You can see that more clearly on this diagram here. And cells reside like this, of course, naturally in tissues. And what happens in vitro, in the laboratory, we deconstruct these relationships and we place cells into vessels such as this petri dish. And cells then grow on these flat glass or plastic polystyrene substrates, most often polystyrene in multiple plates, petri dishes, flasks, and so forth. What happens, of course, is the cell changes its shape. You can see it flattening to adapt to that flat foreign environment. And when that happens, there are changes to the cytoskeleton, remodeling, changes in gene transcription, protein translation, and function. So when studying cells which are flat like this in 2-dimensional culture, they don't behave in the same way as they do in vivo. If we look more closely at this, a cell is almost 50% against the plastic substrate, or it's against the medium. They're flat, very little interaction between cells, and this is really important in signaling processes. This is certainly restricted in vitro, and certainly it's not possible to create more complex structures, as I've mentioned, in vivo. So if you look at an individual cell, you can see how this fibroblast, which has been visualized using a confocal microscope, you can see how the cell has become very flat in 2-dimensional culture, and whereas in 3D, using our Alvetex technology, it's 3-dimensional all the way around. The same applies to the nucleus. See how the nucleus is very flattened here in 2D, whereas in 3D it's spherical. And it's well known in the literature that if you do change the shape of a cell or the organelles within a cell, that leads to changes in gene and protein expression. So this is a phenomenon which is known, but yet it is ignored when people are growing cells in 2D culture. So, excuse me, imagine a technology that prevents the shape change from occurring. So it's a technology that enables simple and routine 3-dimensional culture to recreate a more natural environment. So what we have on this slide is the in vitro situation on the left, and on the right in vivo, and these are actually the same cell type. Here they've been grown in Alvetex in 3D, and here they've been engrafted into a mouse to form a tumor and growing in 3D as well. And they're pretty much indistinguishable. This technology enables you, therefore, to grow structures in vitro which are more representative of real tissues, to enable you to get genuine 3D culture, enhanced viability and function. So what is this technology? It's called Alvetex Scaffold, and it's made from polystyrene. So on this bottom line here, we can see a scanning electron micrograph of the polystyrene scaffold. It's highly porous. It's actually 90% space, 10% plastic, and the space is which the cells grow. So they move into that space and they form structures such as what we see here in 3 dimensions. This is a scaffold which has been sectioned. So it's a transverse section through the membrane. The membrane is 200 microns thick, that's seen here at higher magnification. And it's very thin, which means that cells can move into the material very readily, and the exchange of nutrients and waste products is very efficient. If you think about it, a cell in the middle of this structure is only 100 microns from the source of the medium, either on the top or the bottom. This scaffold is now full of cells, so that's why you can't see the structure, but what you do see is like a slab of tissue-like material which has been produced. So these are the products which are available. So we have multi-well plates, so there's the 12, 24, 96, and also we have well inserts. 6 and 12 well are currently available. The inserts enable you to feed the culture from above and below, as shown here. So it's like a standard transwell, but now we have Alvetex scaffold in here to enable 3D growth. Of course, the plates you have just feeding from above, because it's in the bottom of the plate. In this scenario, we've got a larger volume of medium to support the 3D culture, and that's represented by this device here. So this is a Petri dish, and inside there's a cradle which houses 3 inserts. So you can get a large volume of medium in here to support the 3-dimensional culture of your cells. So that's good for conditioning large volumes of medium, for doing long-term cultures where you don't have to change the medium regularly. Alvetex can be used in a number of ways for cell culture, and this is summarized in the schematic diagram. This is actually taken from our brochure, and it's very versatile. So you can mix 2 cell populations and seed them in 3D. You can do that in the plates or the inserts. You can do 3D culture in the insert and 2D in the plate. So if you're interested in paracrine factors and signaling, you can also do the same, but in 3D and 3D. You can put 2 3D cultures together and simulate layers of different cell types as you would get in tissues. So it's very versatile technology. So if you go to our website, you'll find a very large amount of evidence demonstrating how Alvetex can be used in different applications. So how growing cells in Alvetex can influence the structure and function of the cells, their growth and differentiation, the use of Alvetex to create novel assays and models, and to create quite organized structures as we see here. So let's now move on to the focus of today's webinar, having introduced the technology, that is its application for the growth of neural tissues. So we've had a long-standing interest in mammalian neurogenesis. And what we have here is the embryo showing neuroepithelium, and that's then shown schematically, and in that neuroepithelium cells proliferate, they migrate, and they differentiate. So we've been working with these cells, and these are neuroprogenitors. We've also been working with other progenitor and stem cell populations, and we've been using our technologies developed in the company to grow these cells in 3D and also to differentiate them. So let's now go through some of these data. So one cell type that we've been working with is a human pluripotent stem cell. You see those stem cells here, and we have SOPs and protocols to induce their differentiation into neurospheres and associated neuron populations. And you can use these cells, of course, for research, testing drugs, screening, etc. So here are some stem cells, pluripotent stem cells growing inside the scaffold. You can see them, there's lots of different examples. This one's a nice example. We've got a void here, space inside the material, which is fully occupied by cells. And notice how closely packed they are together. And that's very typical of the stem cell type. We can also then take those stem cells and induce them to differentiate. So what we're looking at here is a transverse section through Alvetex scaffold, and it's been prepared for histology and H&E staining. These are stem cells which have been induced with retinoic acid to differentiate, and we see these neural rosette structures developing inside the material. And these neural rosettes you also see within a teratoma of those same stem cells when placed in an animal. We can characterize these cells by doing immunocytochemistry. So this is staining here for a beta-3 tubulin, and we can see the presence of the neurons very clearly. This is some older work, but nonetheless it demonstrates nicely how stem cells can form neural networks in 3D. So on this slide, we've got evidence of the neural networks growing across the material. We've also got here how we can coat the material with various reagents. So we've used poly-D-lysine or a combination of poly-D-lysine and laminin, and we're then measuring neurite length, and you can see differences between 2D and 3D culture. In this experiment, we took these 3D cultures, or 2D cultures as well. We extracted protein, as you can see, and we then ran Western analysis for various neural markers. And you can see quite nicely that neural markers such as these growth-associated proteins here and here, so MAP2C and GAP43, are regulated at much higher levels in 3D compared to 2D. All these data have been normalized to take into account differences in the amount of total protein and number of cells. So the conclusion is that growing cells in 3D leads to enhanced neurogenesis and enhanced network formation. So that work was achieved using stem cells and inducing those stem cells to make neurons. And classically, one would use all-trans retinoic acid. It's a vitamin A derivative, it's a natural compound, and it's often used in stem cell work, particularly in neuroscience. But one of the issues about this molecule is that it is highly unstable, sensitive to light, oxidation, and temperature, which is a real issue because stem cells are also sensitive to different concentrations of retinoic acid. So if you're uncertain of the concentration, you can get different differentiation responses when using partially degraded ATRA. So this is an issue. So what we've done is we've re-engineered this molecule and created a synthetic version of all-trans retinoic acid. It's known as EC23. And we now use this molecule to robustly and reproducibly induce our stem cells to make neurons. So just to show you, you've got ATRA here on this column and EC23 here, different concentrations. These are stem cells making neurons. They've been stained for TUJ1. Notice that the low concentration, that the EC23 is still very potent, whereas with ATRA, these stem cells respond less. And what we see basically is an enhanced neurogenic response with decreased heterogeneity and increased potency for EC23 over ATRA. So this is the molecule of choice for the work that we are using, and I am presenting with 3D culture from going forward. So what we do is we take our stem cells, we add our EC23. Incidentally, EC23 is a commercially available molecule which you can also acquire from Renovate. We make neurospheres, and then we plate those neurospheres either in 2D or, as I'll show you in a moment, in 3D. And these neurospheres are highly reproducible, and they produce very large numbers of neurites. And you can see those neurites here radiating from the population of neuroprogenitors. Okay, so very efficient. So then what we do is we take those structures, and rather than putting them in 2D, we take the neurosphere and we place it on the surface of Alvetex. And then the neurites radiate out from the neurosphere. Those arrows are actually the wrong way around. So then what we can do is we can visualize the neurosphere on the surface of Alvetex, either from the top, the side, or the bottom, using fluorescence microscopy. So here we are from the top, the side, and the bottom. So you'll see the neurites radiating out, but they're also now going into the material, okay, from the side elevation. And these neurites have journeyed all the way through the scaffold, okay, so they're on the bottom here, and you can see them very clearly, and you can measure and quantify them. So we're using this model to study the formation of the glial scar, and we're interested in how human stem cell derived neurons growing in 3D interact with molecules that are found in the glial scar. So the glial scar is formed when there's a traumatic injury to the spinal cord. So a compression injury, for example, will activate astrocytes, oligodendrocytes and so forth, to produce on their surface these types of inhibitory molecule. And these inhibitory molecules suppress neurite outgrowth. So we're particularly interested in CSPGs here and their role in suppression of human neurite development. So we can develop this experiment. So we take our human neurosphere, neurites growing out, we then take a scaffold, we coat it with aggrecan, and we hypothesize that we'll get suppression of neurite outgrowth. And then if we look at the literature and also what is known about neurite growth, we can predict the use of certain small molecules which will enable recovery and rescue of that inhibition. So things like ROC, things like these retinoid agonists. So let's look at some of these data from this experiment. So in the control, just as before, you've got your neurogenesis on the top, and you've got the neurites going into the Alvetex. And that Alvetex, incidentally, has been coated with laminin. And we also, here in this example, we have laminin, but we also have aggrecan, which is the molecule expressing the CSPGs. So we can study, therefore, the interaction of the neurites with this molecule. And you'll see in this example that neurite outgrowth has been significantly reduced. If we look at the bottom elevation now, so these are the neurites coming all the way through the material, we can see how in the control, neurites as before, but when coated with aggrecan, much fewer neurites. We can now incubate the 3D culture with those small molecules. So this example is for AC261066, which is the RAR. This is a retinoid agonist of the beta-2 receptor, and we see rescue or more neurites coming through. And we can quantify this using a basic digital analysis package, and we can see in the control, aggrecan here, suppression, aggrecan plus the retinoid here, you get recovery, but also with the ROC inhibitor, we get recovery. So it's a nice example of how you can begin to grow your cells in a three-dimensional system and also study the role of important molecules involved, in this case, in glial scar. But we can take this one step further using Alvetex technology, and we can do a co-culture studying how the neurons interact with the glial cells. And this is what we've done here. So we first of all established a culture of glial cells inside Alvetex for 7 days, and then seeded onto the surface the neurospheres. So in the control, you'll see the surrounding black, because there's no other cells there. You'll see the neurites growing out from the neurosphere. In the co-culture, what we see now are lots of DAPI staining nuclei. These are, of course, the glial cells that you're looking at. And in the middle, we have here a neurosphere, and you'll see some neurites growing out. But notice how the neurites tend to grow around the neurosphere and not necessarily radiate out like they do here. So they're being, in some way, inhibited by the glial cells in the 3D culture. So we can do the same again as before. In the control, we can determine the number of neurites going through, and in the co-culture, there are no neurites coming through, but in the co-culture plus, in this case, the ROC inhibitor, the Y27632, we do get neurites then coming through again. So this small molecule is indeed having a positive effect on the rescue of the inhibition of neurite outgrowth due to the presence of the astrocytes. And you can quantify that again, and it's very obvious. So I hope what I've shown you demonstrates that Alvetex influences cell structure and differentiation and function. We can also take primary tissues and grow them directly on the material. So this is actually a piece of embryonic tissue which has been placed on Alvetex, and the cells grow out inside the scaffold. So there's never any 2D growth here. It's primary directly into 3D culture. And we can think about how we can then use that feature as well to support the growth of primary tissues. So this is another way of thinking about using Alvetex. This actually is an embryoid body, so it's a structure which is derived from stem cells and then maintained on the surface of the Alvetex for a longer period of time than one would normally be possible. And we can see neural ectodermal differentiation here in this structure. So this is actually a neuroepithelium. You see that really nicely here. So this is work from human embryonic stem cells. And actually, these structures growing this way, you can see evidence of all three germ layers. And potentially, this is an alternative assay for doing a teratoma and using animals for transplantation experiments. So let's look a little more closely at other primary tissues on Alvetex. So here we have an example of a brain slice culture. So this is work which has been completed by scientists in the University of York. So normally they would take a tissue slice and maintain it on a millicell construct. It's a device with a semi-permeable membrane. Looks very similar to this, and you're able to maintain the viability of your tissue slice whilst you're doing live cell imaging and time-lapse video. But one of the issues is the viability of the tissue decreases over time, but also it moves. So you can see here at time 0 and after 24 hours, the actual tissue material has moved 400 micrometers. And that's a real problem, especially if you're looking at studying cell migration and cell movement within the material. So what we did is we, instead of putting the brain slice on top of the millicell semi-permeable membrane, we were able to use Alvetex, place the tissue on the surface, and the tissue does not move. It moves very, very little compared to what we see up here. So that's a distinct advantage. Plus, also, Alvetex is much more porous than a millicell insert. And that also is very good for the viability of the tissue. So in another collaboration, this is some of the work which is done by a group in Reading University in the UK. They were taking primary cortical neurons from embryonic mice, and they were preparing them and seeding them onto Alvetex. The Alvetex was prepared by, first of all, incubating the scaffold with FBS at 37 degrees for 2 to 3 hours, and then seeding the cells directly onto the material. You can see just from above very clearly the cells, the DAPI, and also the staining here for the neurites. You can see that very nicely. So these cells are very happy to grow in Alvetex, and this group is using this approach for both rodent and human primary cells and are interested in electrical recordings using microarray electrodes from these cells. So just to finish off, go quickly through some methods that are compatible with Alvetex technology. So what we have here is a slide looking at visualization of the culture. So when you're growing cells in 3D, it's much more difficult to visualize them on a standard microscope, standard inverted phase microscope, which you will find in the normal cell culture lab. So to get around this problem, we've developed a very simple protocol where you just stain your culture and with neutral red, and you can now visualize the cells very readily using that microscope. Now, you don't have to do this all the time. It's only in the first instance, if you wish, to get a good idea of cell distribution, viability, etc. So I mentioned very much that we're kind of producing tissues in vitro using this technology, and of course you would then treat the outcome just like you would tissue. So what we've got here are 6 scaffolds at different time points, having been fixed up to 21 days. So then the membranes are embedded. So just like tissue, they're put into paraffin blocks. They can also be put into resin blocks as well for electron microscopy, for example. These blocks are then sectioned on a standard microtome. You can actually see here on the microscope slides the culture appearing over 21 days. And of course, down a microscope, you can visualize structures such as these. So once those sorts of slides have been generated, it's possible to do immunocytochemistry, in situ hybridization, laser capture, and so forth. Some antibodies, of course, don't like paraffin and formalin fixation, so it's possible to do cryosectioning and immunocytochemistry. We have methods for this as well. Confocal I already mentioned. Electron microscopy here, so transmission on the left. So these are 2 cells here at high magnification in Alvetex. They've got this nice cell junction between them. And this is scanning in electron microscopy. This is of a skin cell type growing in the scaffold. So you can see a bit of scaffold here, and then we've got at the top here cells which are beginning to stratify and keratinize. And then, just like standard cell culture plastic, it's possible to coat with various reagents. These are all commonly used in cell culture. So ECM proteins, lysines, matrix gel, pure matrix. And here's some examples. The nice thing about the filamentous proteins is that you get a nice 3D web of protein inside the 3D scaffold. Now, some cells are epithelial in nature and they naturally grow as simple monolayers. So think of some epithelia in the body lining cavities and vessels. So they would not be ideal growing inside Alvetex. So what we can do is we can take Alvetex and we can coat on the surface a collagen gel and then seed onto the surface of that a epithelial cell type. And these are actually CACO2 cells, representative of the intestine, and there's collagen gel, and underneath that is the Alvetex. So the advantage of doing that is you can then think about creating structures such as this. So this is a schematic of the simple epithelium, basement membrane, and lamella propria. And collectively that makes a mucosa. So that involves co-culture, and that's exactly what you can do. So you can, first of all, culture inside the Alvetex fibroblasts, have a gel on the surface mimicking the basement membrane, and then on the surface of that have the epithelial cells. And then you've created a mucosal-like structure in vitro. So what this means is that you've got now more enhanced complexity and physiological relevance. It's also possible to transfect the cells in 3D using reagents developed by Mirus. It's possible to get from the culture protein and nucleic acid directly using commercially available kits. So you can, once you've got your protein, you can do Western or run gels, etc. Various commercial kits for cell viability are available and have all been adapted for 3D use using Alvetex, and there are protocols for many examples online, NTT, NTS, and so forth. It's also possible to determine the number of cells inside a 3D culture based on double-stranded DNA, and this is achieved by using the pico-green assay. So one first of all creates a standard curve, so 2D culture where you can determine cell number and absorbance, and then what you do is you take your 3D culture, you take out the DNA, you measure it on the curve and determine the number of cells. So what I've delivered to you today is a webinar with a specific application in neuroscience, but there are also many other applications of this technology. If you go on to our website, so reinnovate.com, go to our technical resources section, you'll find lots of information about how to prepare the culture and set it up, to analyze and monitor it, and so forth. And all this is available online, as are instructional videos about the technology. And indeed, this webinar is recorded, and you are able to play this back and look at it again online. So I'd just like to finish there and summarize that Alvetex is indeed a very versatile platform for 3D culture. It is very straightforward to use. It's very effective, as the data demonstrates. The Alvetex product is now available commercially from a variety of distributors on a global basis, and it is beginning to be adopted and used widespread. There are scientists who are now sharing their data with us, publishing their work, presenting their work at conferences, and slowly but surely Alvetex technology is being used for advancing research using in vitro methods.